Okay, good morning and thank you for coming. Today I will be talking about the work I did as a postdoc, modeling and inference of eco-evolutionary dynamics in seasonal environments, toxic algal blooms as a model system. Ecology and evolution are very often interacting rather than isolated processes. Because of this, the study of eco-evolutionary dynamics is necessary and important because first, it advances fundamental knowledge in ecology and evolution, and second, it has important implications for ecological conservation and environmental management. Here, we focus on aquatic planktonic systems consisting of phytoplankton and zooplankton. They can be found in a variety of water bodies, including ponds, creeks, rivers, lakes, and oceans. These systems are ideal for studying the ecosystem consequences of eco-evolutionary dynamics for two reasons. First, plankton have a short generation time, which means they can potentially evolve rapidly. Second, these plankton play fundamental roles in food web and ecosystem functioning. My specific focus in this work is toxic algal blooms, also known as harmful algal blooms. An algal bloom occurs when algae in a water body become so abundant that they turn the water body into a green color. These algae also produce large amounts of toxins, sometimes killing other organisms living in the same water body. These algal blooms can occur naturally or be induced by human activities, and they can have detrimental effects on environment quality or biodiversity. Toxic algal blooms are dynamic systems involving an interplay among ecological and evolutionary processes. The ecological processes include trophic interactions among nutrients, phytoplankton, and zooplankton. And the evolutionary processes include genetic diversification of toxin production, as shown by a 2023 current biology paper by Weisskaver et al. Eco-evolutionary dynamics, eco-evolutionary hysteresis to be precise, has been proposed as a mechanism for toxic algal blooms. This explanation is pioneered by a 2016 study in Ecology Letters by Driscoll et al. In an ecosystem, hysteresis refers to an alternation between two stable ecosystem states. In the context of toxic algal blooms, on the x-axis is a toxin production trait or environmental state, and on the y-axis is the abundance of algae. Toxic algal blooms can come and go repeatedly when change in toxin production trait or environmental state drives the system to alternate between a high abundance state and low abundance state. In this work, we take one step forward and show that eco-evolutionary hysteresis is a testable hypothesis for toxic algal blooms. This work is based on a mathematical model developed by Boris Sauterets, a joint lead author of this work, and Regis Ferrier, my postdoc supervisor. The foundation of this model is what we call the ecological theater for toxin evolution. It is a simple MPZ model where the major players are N, nutrients, P, phytoplankton, and Z, zooplankton. The abundance of nutrients may or may not be seasonal depending on input from the abiotic environment. The input from the abiotic environment is also termed environmental forcing. The zooplankton abundance can grow, and this is modeled as a simple logistic growth. The phytoplankton abundance can grow through photosynthesis. Importantly, there's a trade-off between photosynthesis and toxin production. This trade-off is determined by a toxin production effort trait denoted by alpha, which ranges between 0 and 1, and it determines the proportion of the algae's effort devoted to toxin production. Through a process called mixotrophy, the algae produce toxin, killing zooplankton. The killed zooplankton can provide the algae with additional nutrients, which facilitates the growth of the algae. The toxin produced by the algae has another effect, that is to reduce the amount of grazing or competition from zooplankton. We call this process interference. Our ecological theater sets the stage for eco-evolutionary dynamics to happen. Here on the x-axis is the toxin production effort alpha. The system can move along the y-axis through trait evolution. On the y-axis is the abundance of algae, with low abundance corresponding to a non-bloom phase and high abundance corresponding to a bloom phase. And the two stable ecosystem states are shown as two solid black branches in this graph. Starting on the lower branch, evolution of increased toxin production effort causes the zooplankton abundance to collapse. Because of this, the system shifts from the lower branch to the upper branch, and at the same time shifts from the non-bloom phase to the bloom phase. Once on the upper branch, evolution of decreased toxin production effort causes the zooplankton abundance to recover. Because of this, the system shifts from the upper branch back to the lower branch, and at the same time shifts from the bloom phase back to the non-bloom phase. So far, a hysteretic cycle has been completed. I just talked about a scenario where hysteresis is caused by trait evolution. But in reality, hysteresis can be caused by trait evolution, seasonality in the abiotic environment, or a combination of both. So we consider different scenarios based on the presence and sufficiency of trait evolution and abiotic seasonality. We identify a total of 6 scenarios and solve the conditions for each scenario using SymPy, a Python package for symbolic mathematics. Then we develop a hypothesis testing framework using simulation-based inference, which includes 3 steps. Step 1, we simulate ecoevolutionary dynamics using an individual-based version of the model, including all 6 different scenarios. Step 2, we extract summary statistics belonging to 3 categories: ecological, these include the abundances of nutrients, phytoplankton, and zooplankton. Phytophenotypic, which is alpha, the toxin production effort, and phylogenetic, which includes phylogenetic distance between lineages and the total number of lineages. Step 3, we use approximate Bayesian computation, ABC, to test whether simulations can be correctly reattributed to the model that generated them. To evaluate model reclassification performance, we constructed what is called a confusion matrix. Each bar represents multiple simulations under a model, and each color in it represents the proportion of simulations being reclassified as belonging to a model. Our results show that a model reclassification performance is good in general, with each model having an accuracy of at least around 60%. We quantify the importance of each summary statistics as the reduction of reclassification accuracy when that statistic is removed from the inference. We also generate shadow summary statistics by reshuffling each summary statistic randomly. The importance of these summary statistics is then used as a reference against which the importance of real summary statistics can be compared. We also visualize how different scenarios differ in each summary statistic to provide an intuitive understanding of how the inference works. What is shown here is that the more important summary statistics show larger variation among scenarios, where the less important ones show smaller variation. We also simulate 2 different empirical sampling schemes. Summary statistics are extracted at either the beginning of each abundance peak for the algae or at a random time during each abundance peak for the algae. We then compare these results across different scenario combinations and sampling schemes. Specifically, we consider all scenarios versus evolutionary scenarios only. And we find that sampling at a random point during each abundance peak reduces inference accuracy. We also find that the most important summary statistics change depending on scenario combination and sampling scheme, but some general patterns do exist, which I will summarize later. So our main findings. Compared to sampling at a random point during each abundance peak for algae, sampling at the beginning of each peak allows more accurate hypothesis testing. Phylogenetic distance at the second abundance peak for algae consistently ranks as one of the 2 most important summary statistics. Depending on scenario combination and sampling scheme, phylogenetic or phenotypic tends to be the most important summary statistic category. Ecological, which includes the abundances of nutrients, phytoplankton, and zooplankton, is never the most important summary statistic category. We also find that sampling at a random point during each peak most significantly reduces the detection of a scenario which is scenario number 6, where both trait evolution and abiotic environmental seasonality are present, and either is by itself sufficient to cause hysteresis. So some takeaways. First, ecoevolutionary hysteresis is a testable hypothesis for toxic algal blooms. And this hypothesis can be tested through ecoevolutionary modeling and simulation-based inference. And the second takeaway is empirical biologists looking to collect relevant data should ideally target the onset of each algal abundance peak. And the third takeaway is ecological data, which includes abundances, have very limited power to discriminate among scenarios, which means collection of phenotypic or phylogenetic data is crucial for hypothesis testing. There are some future directions. First, I want to see how sensitive these results are to different thresholds for detecting abundance peaks. And also, we would be particularly interested in developing a new mathematical model that more faithfully captures all features of the conceptual framework, a conceptual model of Driscoll et al. 2016. And in particular, we would want the new model. We would want the new model to be set up in a way that reversal of the direction of selection is caused by a switch between what we call public goods and private goods, where the public goods are the individualistic benefit of toxin production, and private goods are the collective benefit of toxin production. With that, I'd like to thank everyone in Regis Feriai's group at the University of Arizona, and also thank the NSF for funding this work. And if there's time left, I'm happy to take any questions. Oh.
Yeah, so you kind of referenced this. Thank you for your talk. I think that was super interesting. And at the end, you reference briefly like looking at public versus private goods, and so which is closely tied into potential cheating in the system. And I'm just curious how you think, just kind of, even though I know you haven't modeled this yet, how cheating might impact the results, where some algae potentially don't produce the toxin, but then as a growth will still benefit from the toxin of other algal strains.
Yeah, thank you for the question. So the question is about, so my understanding of the question is about how cheating, how cheating works like in this model, right? Something. So that's something. So ideally, we would want to build a model where I like to go back to one of my slides, if that's so. So here, ideally, we would want. So here you see that trait evolution goes. So the toxin production effort evolves to be bigger on the lower branch and evolves to be smaller on the upper branch. Ideally, we would want this reversal of the direction of selection to be driven by a switch between private goods and public goods. So private goods is what is similar to what you mean by cheating. And what we think, in the process, we think that can be described as cheating is when each algal individual, they use their toxin to kill the phytoplankton, and only the dead phytoplanktons nutrients can only be used by the individualist, the algal individual that did the killing. I guess I'm out of time, but I guess we can talk more after.
Bonjour tout le monde, je m'appelle Abdel Halloway, je suis professeur à Case-Western University à Cleveland et je vous présente ma lecture. A mutualism parasitism gradient plus control over nodule access leads to trait variation in bacterial plant mutualists. And that's all the French you're going to get from me today, because that's all the French I can handle. So, Before we begin, I want to first acknowledge my collaborators and advisors. So Dr. Katie Heath, who was my postdoc advisor at University of Illinois, and Dr. Liana Burkhardt at Penn State University, and also my funding sources. So this was done with the NSF PRFB while I was at the University of Illinois, and the resources from Case Western. So just want to acknowledge that. So just want to begin with mutualisms. Mutualisms are ubiquitous and important, right? They can be stuff like pollination services, or they can be seed dispersal, right? And they're very important, especially for non-motile organisms like plants and such. However, mutualisms also show an incredible amount of variation. For example, here we have a phylogeny of the legumes, and you can see that in the purple, you have obligate mutualists, so they're obligate fixers. You have facultative fixers in green, and you have the non-fixers in orange. So you see a lot of variation in whether plants can use and to the degree in which they use mutualism. And that comes in two types of variation. You have within species variation, and you have between species variation, right? So you can have, for example, if this is like a community of rhizobia strains, you can have some rhizobia, which you can have different rhizobia attached to each plant, which is represented by the columns. So as you go along the horizontal axis, you're switching between different plants. However, you can also have better, more mutualistic rhizobia that give better benefits, as indicated by the more blue rhizobia versus the more orange. For now, I'll just be focusing on between species variation. And really, there are problems with between species variation, or at least the way we conceptualize it. Because mutualism really leads to a lack of diversity in two ways. One, if you're a species that has a mutualist, you have a fitness benefit that allows you to then outcompete other individuals, right? So I have a mutualist, I have a mutualist partner, I have this fitness benefit, I get to outcompete the other one. So that reduces diversity. Also, mutualism tends to lead to convergence on the same trait, right? So if there is a mutualistic rhizobia, a good one, there's a tendency for the two plants to converge to the same trait to use that mutualistic rhizobia. So then there's a lack of differentiation. So several possibilities have been developed to keep and maintain variation. One is to tie mutualism to the environment. So basically in different environments, you will see different mutualists. So as salinity changes or as temperature changes, you'll see differences in mutualists. Or you can allow cheaters in the population, which adds this predator-prey dynamic. However, those still rely on kind of an external source, right? You have to use the environment, or you have to use a kind of predator-prey model. The question that we have is, can mutualistic interactions intrinsically create diversity? So we kind of look to other systems, right? And one key aspect of diversity is the need for a push, right? You have this tendency towards stabilizing selection, positive frequency dependence in mutualisms, but you need something that creates a divergence, right? You need some kind of competition or predation or something that pushes them across. Essentially, species must repel each other. And if they have negative interactions, that could be a possibility. And so one of the possibilities is that you're very mutualistic with the partner, right? So the plant and the rhizobia on the left are very mutualistic. However, the plant and that other rhizobia on the right are very antagonistic, and vice versa. And that creates a repulsion that allows for stable coexistence, right? This may occur through something like a mutualism parasitism gradient. And you don't necessarily need a parasitic gradient, as I will explain soon, but you need some kind of like negative fitness benefit. So a lot of this work, as maybe you have caught on, is inspired by the microbial mutualism, whether it's mostly rhizobia or what do you call it, mycorrhizal fungi. But basically in this mutualistic interaction, the benefits are nutrient trade. Plants get nitrogen, microbes get carbon, and a stable environment. Sorry, I did not know there were timings on this slide. So I mean, you guys got a sneak peek, right? So yeah, there you go. The rhizobia also have this stable environment in which they can reproduce, which is the nodule, right? And wow, so you guys are really getting a sneak peek. However, there are costs associated, right? So there's trait-associated costs, right? You know, production of carbon or production of nitrogen. Also, there's a cost to the host, right, that houses the mutualist in the fact that they have to build these new nodule structures, right? And so that they have to invest a fixed cost into the mutualism, right? Regardless of whether the rhizobia is actually a good or bad partner. And then so there's also a mutualistic interaction where there's a sort of like lock and key model where if you have a nod factor and a nod factor receptor and they match, that allows access and creates the kind of interaction. So maybe you're crazy like me and you like to look at these equations and say, wow, that's amazing. I can, you know, kind of understand it, right? But if you're not, then essentially what's going on is we create this idea that there's a benefit to matching traits, right? So plants have a sort of trait, you know, space. Microbes have a trait space, and if they match in the trait space, right? So for example, these are just numeric values, but if a plant is 3 and a microbe is 3, then you get maximum benefit. However, if you're different, it leads to worse benefits, right? So, you know, like, and it never really goes negative, except when you input the fixed cost, right? So the cost of building, for example, a nodule, right? So if the host now has to build a nodule, then it reduces it and it actually gets a negative benefit, right? Because it creates this cost for a bad partner and then it doesn't get any benefit. So now it has negative fitness based upon this cost. Now we add one more key thing, which is we allow control over access or association, right? And basically the idea is that if you are, say, a plant with trait, you know, 3, then you allow those that are very similar into the nodule and you don't allow those that are different. And that doesn't really change anything for the microbe. We also have that for the microbe. It doesn't really change its fitness benefit. However, it does change it for the host, right? Because if you see in the host, it has a kind of squiggly line, right? And so if you are close, if you are within that range, that leads to evolutionary convergence, while if you are outside of that range, that leads to evolutionary divergence. Plants don't want to evolve towards microbes that are bad for it. And so basically, we see the exact same thing, right? When these traits start very close, we see evolutionary collapse to just a single trait. However, if they start far apart, then we get to see the divergence, right? We see these host plants, they evolve towards their respective microbes, and then now the systems are moving away from each other. So that creates very isolated, you know, plant-microbe associations. So thanks to especially Liana, she had run a greenhouse experiment that looked at selection with microbes. And essentially what it was is an experiment for host preference for microbes. So there were, you know, about 15 metacargo plant species and 178 cynorhizobium strains. And essentially the experiment was that picture over there. You start with an initial I just want to also say that half of those cynorhizobium strains were one species, Medicae, and the other half were Melilodi. So you start with an initial community of cynorhizobium, you give it to a plant, and then you see how those frequencies change, right? So you see, for example, for A17, those top 3, what do you call it? Cynorhizobium strains increase in frequency, right? So the plant is selecting for those. However, the bottom ones are less than what they started out with, so they're decreasing in frequency, right? So we can see what the preferences are, right? So all plant, and the thing we also do is all host plant species or varieties are giving the same initial frequency, so we can track it that way. And yeah, and then we see the change in frequencies over a certain period. And with that, we can actually create an interaction network, right? And we get a network that kind of looks like this once we kind of analyze it. So we have the plants, the host plants on the vertical axis, and then we have the microbes on the horizontal axis. And you can see, so we first, in this network, we don't account for negative selection, right? So we just have positive selection, right? They're just the preferred microbes. And so you can see that there's about 5 different modules, right? Sorry, I just had to point out that tiny one over there, right there. But it's about 5 or so modules. Sometimes when you do the analysis, it's maybe like 3 or so, but that's what it is. And you can see that they are very different. So for example, if you look from pro to mirror, not including mirror, pro to mirror, they only associate with Melilodi for the most part. Melilodi only associates with those species, or selected. And then if you look at the other ones above mirror, then they're really associating with Medicae, right? Maybe a little bit of Melilodi are in that module. And then mirror has a little bit of both, but it seems like they're very separate, very isolated. And so we have these 5 different modules. We can calculate a kind of modularity score, right? So if we look at the proportions of these connections in the modules, weighted by the connectivity, we can see like, okay, how many connections are actually in the modules versus just like kind of scattered around, right? Once again, for all the people who like the math, this is what it looks like. And we get a modularity score for this particular network of 0.7, right? Now, what does that mean? Well, nothing unless you compare it to a null model, right? So we have 2 types of null network generation. The first one just shuffles the connections, but keeps the modules of the original network, right? So we have the modules of the original network, and then we calculate that modularity score. And then we shuffle the connections to generate these new networks, but then each new network is now then reassigned its own modularity, right? Its own modules. And then we calculate the modularity score. And what we find is, yes, in the first one you have this like less modularity, but regardless, our score is very much on the other side of the 2, right? Suggesting that this network is much more modular than what you would expect from random. And so I just want to go back to the simulations quickly. I haven't done the analysis of like simulating a whole bunch of runs and seeing how they compare. But for example, if we start with many different strains, we can see this kind of collapse, right? Where we have these 2 plant species, they diverge from each other, but you see them sucking up all these kind of plant associations. So this, in our model, has a tendency to increase the modularity of the network, which is something that we see with this real data. So just in conclusion, right, the fixed cost of maintaining a mutualistic interaction can really help in maintaining, the fixed cost of maintaining the mutualistic interaction can create this mutualism parasitism gradient, which can drive mutualism coexistence. However, only if at least one partner can control the axis, because you need that kind of squiggle, right? You can't just have a straight Gaussian. And so the resulting community creates a highly modular structure that is seen in real networks. Now, there are other missing but key features from our model, right? This model is very deterministic, right? Well, so real networks aren't as modular as the one we showed, right? In our simulations, the modularity we would expect to be very close to 1, right? It's still 0.7 in the actual network, and we think that's because there's non-deterministic processes, right? Stochastic variation and all of these things, right? It may be that there's a small cost to mutualism maintenance, right, which may not drive selection as strongly as if there are large costs, right? There's no correlation between the benefits, sorry, that should say benefits and access, right? We assume that there's a correlation in how benefits and access line up, right? That might not be there. There's the possibility of cheating, right? That creates cyclical dynamics because it's a predator-prey interaction, and that should induce variation in the system. And so further analyses would be to analyze the network modularity for all our different scenarios, so like do more model runs and see how likely is it to resemble this real network. And also, so compared to the real data. but also just to get a better ecological understanding of the system, right? This is a greenhouse experiment. There's a bit of artificiality. I'm a modeler, right? So I know a lot about artificiality, but there's a bit of artificiality because you're taking all these strains from everywhere and combining them with all these plants from everywhere. It's a question of like, well, do they actually interact? So having a better geographic understanding, ecological understanding through the geographic distribution should allow us to, get it to a better ecological understanding of what's going on in the system. And so with that, I just want to say thank you, and just, yeah, of course, I want to say thank you, and if you're interested in postdoc or, PhD stuff, I just started, it's going to be a year and a month on Wednesday or Thursday. So very fresh, still building my lab. Feel free to reach out to me, maybe not today because I actually have a train at 12 o'clock, so I'll probably be going very soon, but feel free to reach out. Thank you.
Okay, thank you all for coming. My name is Johnny. I'm a PhD student at the University of Edinburgh, and today I'm going to be talking about some of the first work in my PhD on dental calculus in mammals, diet, phylogeny, and the use of museum specimens. If you like pictures of skulls, you're in luck. So I'll get started a while ago with Heraclitus, who said there's nothing permanent except change. And indeed, the planet and all life is in a constant state of flux and change. And this change can, of course, be evolutionary change, as we are all well aware. The effects of anthropogenic impact, arguably the most pressing of which is climate change. And with shifting perspectives of interconnection, we're understanding the interconnectedness of the world and the interconnectedness of ourselves, especially things like the microbiomes that organisms support and the interactions that the microbiomes have with their hosts and the diverse effects they can have on them. So with a hologenomic perspective, we're able to link the macro and the micro. And so this, of course, begs the question of how can we study these host-associated microbiomes, and especially how can we study microbiome changes through time. So, to that end, I would like to introduce to you dental calculus, which is a mineralized plaque that forms on teeth. You can think of it like a fossilized oral microbiome. So as material in the oral cavity sort of settles and forms a biofilm and calcifies in layers and layers and layers, it traps oral bacteria, proteins, and food remains. And this allows comparisons across wide timescales as the dental calculus can be preserved for a very long time. And just a note on the oral microbiome, particularly interesting as it forms the bridge between the host and the environment, so it can be a point of first contact or initial colonization of the host by environmental microbes. And dental calculus has been used primarily in the past archaeologically in humans to look at things like diet using phytoliths or plant remains. But it has been used increasingly with ancient DNA to answer diverse questions. So some of the things that I mentioned earlier, evolution and the changing ecology of the oral microbiome in African hominids, or the effects of anthropogenic impact. So this study looked at the effects of antibiotic use on the environment using dental calculus in bears, or even local adaptation, where it was found that the local ecology of mountain gorillas is reflected in their oral microbiome. So clearly there's huge potential and a diversity of questions that can be addressed here, but relatively few species have been studied so far. So this, of course, leads us to wonder, where else can it be found? And this is the first big question I'd like to discuss. Which species develop dental calculus? And then following that, what are the factors that lead to dental calculus formation? And then using that, how can we inform future collections-based research using dental calculus? So to do this, we conducted a survey of 5 museums: the Natural History Museum of Vienna in Austria, the Royal Belgian Institute of Natural Sciences, and the Royal Museum for Central Africa, both in Belgium, the National Museum of Scotland, and the Natural History Museum of London. And we surveyed over 1,700 specimens of 143 species, and we were able to get at least one species from all mammalian orders that have teeth. So what we did is had a scoring system of 0 at the top, so no calculus, going up to 3 at the bottom, which is a sort of complete incrustation. And here I have some phylogenetic breadth, different diet types, and you can see that dental calculus can present itself in many different ways. So perhaps you're wondering, how do we know that what we think is dental calculus is in fact dental calculus? So we sampled some of it, and important thing to take away here, the pink and the red are reads for taxa that belong to oral microbiome. So we're seeing an oral microbiome signal in these species that we sampled from, and so we can be more confident that what we think is calculus is calculus. So some preliminary results from the survey. There's a lot going on here. For each species, we aim to get at least 10 specimens. The bars show the diet, so red carnivory, the dark green, the light green are frugivory and herbivory, and then the colored shapes at the outside are whether or not that species had dental calculus on each of the 3 tooth types. So straight away you can see that dental calculus is actually quite abundant across all of these species. 77% of them had calculus, and molars most consistently showed calculus. So if a species developed dental calculus, we found it at least on the molars, if not the other teeth as well. And already, perhaps you can see a link between the diet and the presence of dental calculus. So most of the carnivores have less, whereas the herbivores have more. But there are conflicts in the literature about the effects of dietary carbohydrates and proteins or other things like the pH in the oral cavity, other different minerals and stuff. And I'll get more into the diet in a moment. Because there are also morphological factors that can affect the development of dental calculus, particularly things like the tooth morphology, providing surface for material to adhere to and calcify, the shape of the oral cavity, and then things like the position or size of the salivary glands, all of which range a lot across mammals. And we think that these factors will largely be driven by phylogeny. So I'll begin with talking about the diet. We were able to get diet nutritional composition for all the mammal species that we surveyed. And this was partitioned into 3 groups. Proteins and fats for carnivory, and that's at the bottom of the triangle. Fibers for herbivory, as you can see. And then sugars and starches for more frugivory on the other side. And each one of those dots is the species that we surveyed, and then the color dark to light from low calculus to high calculus. So you can already kind of see the way that they spread themselves off mostly across the protein axis. And we did some modeling to identify which factor is the most important. Since we had compositional data, we worked with log ratios here, and so what we found was that the ratio of protein to fiber had the strongest effect. So less protein, more fiber, more calculus. In addition to that, we also trained a random forest classifier on the diet data. And the purpose of this was to get sort of a simple single rule. So with a minimum information about the diet of the species of interest, you can make an educated guess as to whether or not they would present dental calculus. So if you overlay those dietary cutoffs that we extracted from the tree ensemble on the figure, you can see that it mostly captures where you cannot find calculus. But there are some outliers. And these are unaccounted for by diet composition alone. So, in addition to the diet, after accounting for it in our modeling, we found that there was some additional variance explained by the phylogeny. So, we looked into the presence of phylogenetic signal using several measures. I'll talk first about the mean pairwise distance. So, this is the average distance between all pairs of species that had calculus across the entire tree. So, you can see that the observed mean pairwise distance is about 2 standard deviations lower than what it would be if the trait was just randomly distributed. So we seem to have some level of phylogenetic clustering at a deeper scale. Whereas with the mean nearest taxon distance, which is the average distance from any species with calculus to its nearest neighbor with calculus, it doesn't seem that different from random. And we think that this might reflect some recent dietary changes. So 2 examples here, the binturong and the macaque. You can see there's been, relative to their nearest neighbors, a change in their diet. Mostly a decrease in carnivory with the binturong, an increase in the macaque, and then accompanying changes in the dental calculus scores where we're seeing less calculus on some of the other teeth. So we think that this phylogenetic signal can relate to some of those morphological things that I mentioned earlier, such as the position of salivary glands or tooth morphology. Here a seal tooth, I believe that's a panda, so you can see the really significant differences between the 2. So I'll just pull out some examples, starting with the bovids, which consistently very herbivorous and consistently have calculus. Just a quick note I didn't mention before on the big figure. The lack of a shape entirely means that the specimens we surveyed didn't have enough teeth for us to score them. So it doesn't mean that they didn't have calculus there, just they didn't have those teeth. So really there's consistent calculus across the bovids. And we also know that bovids have large parotid salivary glands under their lower jaw. And we found significant differences, small effect, but still significant between the upper and lower jaw when looking at just the bovids. The rodents are another example here. So you can see on the molars that below, well, it's flipped upside down, but below the gum line, there's really strong incrustation. So we know that whatever they're eating, they definitely can develop calculus. And also that they have very large incisors, so that would potentially provide ample surface area for the development of calculus. But that constantly growing tooth pattern that rodents have means there's less time for accretion. And what we found was, indeed, the incisors are significantly less calculus presenting than the molars. So lastly, we had diet data for many mammal species. So using our model that incorporated the diet and the phylogenetic relationships, we made predictions across the roughly 4,500 species that we didn't assess. And this figure here, each tip is a family. The bar graphs is sort of an average diet composition. And then the box plots show the distributions of predicted calculus scores. So we'd hope that this kind of information can be used for researchers to plan studies and access collections efficiently or assess study feasibility if interested in using dental calculus. And also provided to curators to help them identify areas of pressure in collections and stay informed about developments in collections-based research. A lot of the curators that we spoke with and worked with were excited about the potential use of dental calculus, anticipating a lot of interest in their collections and kind of trying to stay ahead of that and be informed before they start to let people in, because the sampling is somewhat invasive. So there are conflicting priorities between the researchers and the museums themselves. So I guess lastly, we had some great symposia on the use of natural history collections. And so I'd like to raise some questions of what the purpose of a museum is. These collections that we worked in were amassed mostly in the 19th century with an angle more towards use of morphology for taxonomy and systematics. Now as technologies change, what we're able to do with these collections change. So what is the purpose of a museum and a natural history collection moving into the future? So that's all. I'd like to thank especially Markella, our honor students as well, that helped us with our surveying, and the curators at all the museums that we worked with. Thank you. Any questions? There's no bell. It's not ringing. Okay, well, that's 12 minutes, so.
So I guess the hypothesis is like recently diverse species could either be very similar in traits and habitat, or because of competition, we have this sort of similar from each other, trying to like.
Sorry, I just missed what you said with the clapping.
So I think there are two hypotheses for like similar species or sister species, right? They're either very similar in traits because of the recent ancestry, or they're very different because they face strong competitive pressure, so there's strong pressure to diverge. Do you think dental calculus could be used to test that hypothesis in any kind of way?
I think it would depend, because with the molars it was really consistent across everything. I don't know that that could be used, but like those examples I pulled up where we did see differences in the incisors and the canines, it potentially could be, and we assume that that reflects dietary changes, but that's not necessarily the case, as it could be some of those many other factors. So yeah, I would say, in short, I guess it depends on the species that you're looking at, and then, yeah, what diverges from their neighbors.
Okay.
Is that?
Yeah, no.
Okay, thank you.
Hi everyone, my name is Kaitlin Allen. I am a postdoc at the University of Florida. And today I'm going to be talking about a project that we're finishing up on niche evolution across highly reticulate phylogenies, particularly in the allopolyploid African clawed frog, Xenopus. And if you'd like a little primer on polyploidy before we get started, it's essentially a whole genome duplication. This is a phenomenon that is really common in plants, but a lot rarer in animals. There are a couple of fish, insects, and a smattering of frogs that show this trait. And there are two main pathways that an organism can take towards polyploidization. One is through hybridization between two species. So you see up there on the left, that organism then has the full genomes of both of the parental species within it. And autopolyploidy, which is genome duplication within a species. So then the resulting offspring species would just have double the amount of essentially the same DNA. And if you're really interested in polyploidy, but you don't know a lot about it, or you want to teach it in your classes, we actually wrote a series of modules aimed at upper undergraduate level students, which are available for free on Cube. So check those out, get a little publicity for those. Studies on plants have been showing essentially that allopolyploid organisms tend to have really high rates of niche evolution. And this can be for a couple of different reasons. Newly formed polyploid species are often immediately reproductively isolated from their parental species. Not always, but they often are because of different chromosome counts, essentially. They have lots of new genetic material to work with. And then because they are the result of hybridization, they geographically overlap with their parental species. So some ecological or geographic separation is usually beneficial for these offspring species. Three main patterns of niche evolution have been observed. Niche expansion or contraction, niche shift, which is kind of an expansion on one side and a contraction on the other, and niche novelty, which is a completely different niche from the parental species. And what we're working with is the African clawed frog xenopus, which is an allopolyploid. This is their phylogenetic network, phylogenetic tree. It's extremely complicated. This is a very complicated group. There are 29 species. Only one of them is diploid. There are 17 tetraploids, 7 octoploids, and 4 dodecaploids. Almost many of these allopolyploidization events are independent of each other. And there are, in fact, 2 different subgenera, Xenopus xenopus, which is most of this really large group on the top, and Xenopus silarana, which is on the bottom. And these actually have independent diploid ancestors, but only one of those diploids is known. And what really got us interested in this project is that when we mapped the distribution of the ploidy levels across this group, we noticed a clear geographic pattern. Essentially, octoploids and dodecaploids are highly associated with the montane areas of Central Africa, while the tetraploids and the one diploid are found in more lowland, widespread areas. There could be a couple different reasons for these patterns, and these could both happen at the same time. But essentially, species with higher ploidy levels may be able to adapt to montane environments more readily, like we talked about with all that extra DNA. Or kind of alternatively, changes in species distributions during the Pleistocene or the Miocene might have allowed polyploidization to occur in the mountains as species kind of moved up and down and interacted with each other through hybridization. So we wanted to see what the patterns of niche evolution in this group were. First by evaluating the frequency with which polyploid species encountered changes in niche breadth, niche shifts, or niche novelty, by constructing ancestral state reconstructions, but also we wanted to look at the habitat and geography for this group to see if the changes in niche that we see are coincident with what we know about their habitat and geography through time. We faced a number of different challenges at the beginning, particularly that a lot of niche evolution analyses actually focus on the realized niche instead of the fundamental niche. So the fundamental niche is essentially where species could live with no outside factors, and that's going to be what's kind of heritable and passed on, kind of a trait of a population or a species. And many ancestral state reconstruction programs can't account for the kind of phylogeny that we're looking at with all those reticulations. So to get into this, we combined niche characterization that can account for uncertainty, so to account for the fact that we don't have the full realized niche or the full fundamental niche, with ancestral state reconstructions that are made for reticular phylogenies. So I'm going to get a little bit into the methods now. We use 4 variables: mean annual temperature, temperature seasonality, annual precipitation, and precipitation seasonality. These are biofilm variables, so pretty broad scale. We delimited an accessible area for each species around only genotype localities, and that's because all xenopus really look almost exactly like that one in the corner, just kind of lumpy little jobs. So they're hard to tell apart, and there's 30 species. So within that accessible area, we binned each climatic variable into a spectrum of niches using the package Addichiball, and scored each bin as present, absent, or uncertain. We created a phylogenetic network using a multi-labeled RAG1 and RAG2 tree that was previously published in a different study by one of our collaborators. We converted that into a network and ran our ancestral state reconstructions using the maximum likelihood-based approaches in the package phylonetworks. networks. We scored the niche change on each branch and mapped it onto our tree. That's the same tree, it's just kind of stylized for readability, if you could say that. So essentially, if a niche was larger than the ancestral niche on one side or the other, then that's an expansion. If it's smaller on one side or the other, that's a contraction. If it's smaller on one side and larger on another, that's a shift. And each of these colors corresponds to how they're arranged on the tree. And if this is a pretty common scenario, where it's unknown on one side, that's the gray, then expanded on the other side, then we count it as an expansion. It could be a shift, but to be conservative, since we don't know what's going on, we count it as an expansion. We ran this for all 4 of our variables, and I'm going to sum up the patterns that we see here in a couple slides. We have mean annual temperature, temperature seasonality, precipitation, and precipitation seasonality. We also ran our habitat and geography reconstructions. And there's a couple of kind of major patterns here that we'll get into a little bit later also, but essentially there are multiple transitions from rainforest to savanna habitat within Xenopus. One with the shift from Silurana to the Xenopus between the 2 subgenera. There's a shift from rainforest to savanna, and then back into rainforest with that large and highly reticulated group at the top. And then geographically, that kind of tracks with the geography of the group, where we see a shift from West Central Africa to East Africa, from Silurana to Xenopus, and then back into kind of West Central Africa with that reticulated Amiotti group. And this actually tracks with what we know from the fossil record, so that does match with what we know about the species. And here are some major niche evolution patterns that we saw across our entire tree. In general, niche contraction and expansion were the most common factors. Niche shifts were rare, and we didn't see any niche novelty the entire time, so there weren't any cases where the offspring species was completely different than the parent species. The different climatic variables that we used showed really different frequencies of niche evolution, particularly that the frequency of niche evolution was much lower in mean annual temperature than in the other climate variables, and those were mostly contractions. And we're guessing that that's because of kind of physiological limits on the ecology of the genus, where everything else can be a little bit looser. Kind of contrary to our expectations, we found that the frequency of niche evolution was actually higher for species formed by bifurcation, so like your traditional kind of split species splitting, than by reticulation, which is the hybridization. And while that's a little bit surprising to us at first, it actually could be a couple different reasons for that. One is that these bifurcating lineages are already tetraploid, so they already have a lot of DNA. The bifurcating lineages are also generally much older. All the hybrid species are quite a bit younger, formed within the last couple million years. And we had a little bit of difficulty characterizing the niches of many of the polyploid species because they're really high elevation, small range species, so they had a lot of unknowns in their niches. But despite all of those kind of challenges, looking at the octoploid and dodecaploid species, we actually were able to see that all of those species, all of the higher higher ploidy species, showed niche divergence for at least one climate variable from one or both of their parents. So still really high levels of niche divergence, kind of when you take all of the climate factors into account. When we put this in a geographic context, we see the highest levels of niche evolution in species with the most extreme ranges. So in particular, Xenopus laevis, which is one of the model organisms that people work with kind of in the medical field. It's found in South Africa and has really wide and really divergent niches kind of across all of the variables, as does Xenopus fishburgi, which is the only one found in the Sahel. There were a couple of different hybridization events that we were really interested in looking at. First, that there was a high elevation group of tetraploids in East Africa that hybridized with a group of tetraploids in lowland Central Africa twice. We were able to characterize the niches well in one of these groups, a high elevation East African octoploid group, which, as expected, showed expansions into lower temperature and seasonality across the most variables compared to the parentals. And then these octoploids, again, hybridized with the lowland Central African tetraploids to make 3 independent dodecaploid lineages. And these, in general, actually showed fairly contracted niches compared to the parental groups. And in the high elevation dodecaploids, we saw expansions into lower seasonality across those higher elevation areas. So to sum up, this is the first study of its kind on a polyploid vertebrate group. And we did see extremely high levels of niche evolution across the genus, particularly, really across the entire genus, both across the ones that are forming through bifurcation and the ones that are forming through hybridization. And this is really strongly in contrast to what we've seen in other studies of this kind using similar methods on diploid organisms, which have shown widespread niche conservatism, even in globally distributed taxa. So similar to plants, our highly polyploid frogs might be doing really interesting things with their niche evolution with all that DNA that they are able to use to their advantage. So with that, I would like to thank, this is a huge project and a lot of people were involved, and I'd like to thank everybody involved. And I would be happy to take any questions.
So I guess I have just one small brief question. I know you're focused on the octoploids and tetraploids, but I noticed that the diploids were very restricted to just like coastal West Africa.
Yes.
What do you think is causing that?
That's an excellent question. It's a little bit hard to say because there's not a lot to compare it with. The only diploid that's known is Silurana, Silurana tropicalis, and there's only a couple other species in Silurana, and they're all found like very kind of coastal Central Africa. Like they don't have that like wide range of habitat that they inhabit, like the Xenopus do. It would be best if we could find really the diploids for Xenopus as well, and actually be able to do a proper comparison across those parentals to offspring, like the original parentals to offspring groups. Yeah, thanks.